Hello guys, I am Harun and this is my YouTube channel Harun Biozone. Now in this video is the second video of my NTA NEET series 2024. This video is based on class 12. Here I completely described only 20 MCQ questions. By these MCQs, I will try to solve important theory portion of your syllabus, from where the questions are coming in all medical exams and also another competitive exam, life science based competitive exam like NEET, gipmer, AIIMS, UVET and School Service Commission recruitment, etc. So watch this video very carefully, that help you a lot to clear the concept of theory. So let's go and enjoy this video. If you like it, please like and subscribe my channel. So let's go and start to solve the MCQ questions. So the first question is, which of the following statement is correct? First, clistogamous flower are always autogamous. So here two terms are present, clistogamous and autogamous. So clistogamous means the flower that present at bud condition, or the flower are closed, we do not open. This is called clistogamous flower. And autogamous means this is one type of pollination where self pollination occur in flower. Okay. Number B is genogamy. Genogamy occur only by wind pollination. What is genogamy? Genogamy is a cross pollination where the pollination occur between two different flower of different plants. Okay. Number C, chasmogamous flower do not open at all. No, chasmogamous means the open flower where the floral reproductive part like androecium and gynoecium are come out from the flower, Number D, geitonogamy involves the pollen and stigma of flower of different plants. So what is geitonogamy? Geitonogamy is one type of self-replication where the self-pollination, where the pollination occur between two different flower of same plant. So here right answer is A, that means clistogamous flower. This is the right answer. So let me explain how here I represent some flower. This is clistogamous flower. This clistogamous flower means they are in bud condition, they are not open. So floral part, reproductive part do not come outside. And this is chasmogamous flower, means open flower where different floral parts, especially reproductive part, androecium and gynoecium are coming outside of the flower. And this is type of pollination. Here two type of pollination occur, that is autogamy and genogamy. Autogamy means when the pollination occur within a same flower, that is called autogamy. So here the flower is bisexual, means both male and female sex are present. When geitonogamy occur, the flower are different but plants are same. That means the plants bear both male and female in different flower. And this is genogamy, means when pollination occur between two different flower of two different plants. This is called genogamy. Okay, so, clear? So here the right answer is clistogamous flower. Clistogamous flower are always autogamous. A number is right. Next question. Select the correct option. Select the correct option. Option describing gonadotropin activity in a normal pregnant female. Gonadotropin activity means the release of different type of gonadotropin hormone, like human chorionic gonadotropin hormone, like luteinizing hormone, like follicular stimulating hormone, that help to regulate the pregnancy period. So let's number A, high level of FSH and LH. High level of FSH and LH stimulates the thickening of endometrium. Here says when level of luteinizing hormone and follicular stimulating hormone level is high, that stimulates the thickening of endometrium. Endometrium means the inner wall of the uterus. Second number, B number, high level of follicular stimulating hormone and luteinizing hormone facilitates implantation of the embryo. Means when the high level of luteinizing hormone and follicular stimulating hormone level is high, that help to implantation or the settling of embryo or zygote into the endometrium wall. That is called implantation. High level of HCG stimulate the synthesis of estrogen and progesterone. Here says when the level of human chorionic gonadotropin hormone that secret from the ectoderm of the embryo, okay, ectoderm of the embryo, and from ectoderm there are two layers present, cytotrophoblast and syncytotrophoblast. So this trophoblast produce the placenta. Placenta attaches the embryo to the mother womb. This placenta secrete human chorionic gonadotropin hormone. So when the human chorionic gonadotropin hormone level is high, then stimulate the synthesis of estrogen and progesterone. Estrogen and progesterone hormone mainly maintain the pregnancy of a female. And high level of HCG stimulate the thickening of the endometrium. The high level of human chorionic gonadotropin stimulates the thickening of the embryo, endometrium. The here right answer is C. When in human female body the human chorionic gonadotropin level is high, then that stimulate the secretion of estrogen and progesterone, and this estrogen and progesterone maintain the thickening of the endometrium wall. So let me explain properly. So what is the function of human chorionic gonadotropin? Human chorionic gonadotropin is a glycoprotein. Is a glycoprotein hormone produced during pregnancy that is made by the developing embryo. Okay, during pregnancy when the embryo developed, these glycoprotein hormone are released. After the conception means attachment of the embryo to the trophoblast, to the endometrium wall, okay, the syncytotrophoblast. Some level can first be detected by a blood test after 6 to 12 days after conception, after implantation of the embryo. 6 to 12 days after implantation, the urine test about 12 to 14 days after conception. The 12 to 14 days after conception, the human gonadotropin are found in blood and urine of the pregnant woman. And in general, the HCG levels will double every 72 hours. The level will reach its peak in the first 8 to 11 weeks of pregnancy and then will decline and level of for the remainder of the age. So यहाँ पे क्या बोला कि HCG is a glycoprotein type of hormone that produced by syncytial trophoblast of the conception. Okay, and after 12 to 14 days, the level of the HCG increase in blood and urea of the pregnant woman, and every weeks the level of HCG increase in double concentration in blood and urine. But after, here looks the graph, after 10 to 12 weeks the HCG level become decrease. But this HCG stimulates the secretion of prolactin and progesterone and estrogen hormone. This progesterone and estrogen hormone maintain the pregnancy, maintain the endometrium wall of the pregnant woman. Okay. The high level of HCG stimulate the secretion of estrogen and progesterone. Estrogen secrete from Graafian follicle. Estrogen secrete from Graafian follicle and progesterone secrete from corpus luteum. Next question. The test tube baby program employs which one of the following technique? So what is the meaning of test tube baby? Test tube baby means when the sperm and eggs both are collected from different source and they fertilize outside of the body in the laboratory. Then 2 to 8 cell stage it transfer into the female uterus or fallopian tube. That is called test tube baby. So here in first option, first A option, the intracytoplasmic sperm injection, intracytoplasmic sperm injection, ICSI means here the sperm is directly transferred into the cytoplasm of the egg inside the ovary through microinjection. So here only gametes are transferred, not zygote. That is not test tube baby, because normal fertilization occur inside the human body. Intrauterine insemination, that means in the uterine portion of the uterus of the female embryo or female body, the sperm are transferred. Gamete intrafallopian transfer. Gamete means sperm. Intrafallopian means sperm transfer in the fallopian tube of the female body. But here zygote intrafallopian transfer. Here the egg and sperm collect from the different source, then fertilize in laboratory, then transfer into the female body. So here the right answer is zygote intrafallopian transfer. So here some explanation. This is the technique of in vitro fertilization. So the test tube baby program is called assisted reproductive technology, ART technology, assisted reproductive technology. The assisted reproductive technology divided into two types. Number one, when the gamete is transferred, and number two, when zygote is transferred. So when the gamete is transferred, this is again divided into GIFT, gamete intrafallopian transfer, IUI means intrauterine insemination, and ICSI intracytoplasmic sperm injection. Okay. In these three conditions, the sperm or gamete is transferred into the female body in gametic condition or haploid condition. But in case of zygote condition, where the test tube baby occur, means the egg and sperm collect from different source, then transfer into the female body. Here IVF, in vitro fertilization. In vitro means outside of the body. In vivo means inside the natural habitat or inside the body. Zygote intra-fallopian transfer. After fertilization, the zygote are transferred into the fallopian tube. Okay. IUT, intrauterine transfer. Here the zygote transfer in the uterus of the cell. So, what is the process of IVF? First, MOIT, multiple ovulation embryo transfer. Normally, in female body, only one egg produced in one ovary in each every month. हर महीने एक ओवरी में only one egg ready होता है, produce होता है. But when different hormones, gonadotropin hormone like LH, FSH are injected into the female body, both the ovary produce many eggs at a time. These eggs are collected. Okay, here these eggs are collected by through micro injection, and sperm collect from male body. Okay, then sperm and egg are fertilized in laboratory outside of the ovary. Then the zygote is transferred into the uterus, either fallopian tube or uterus of the female body. Then the pregnancy is occurred. Okay, so I think this question is solved. So here the right answer is zygote intra-fallopian transfer. This is the right answer. This is the right answer. Next question. Given below are four methods. Given below are four methods and their mode of action in achieving contraception. Contraception means control the pregnancy or of the pregnancy by use of several different natural and synthetic methods. Select their correct matching from the four options that below. So here the pill. So what is pill? Like I pill, like unwanted 72, like unwanted 72, like malady, etc. The pill mainly contain progesterone hormone. Some pills contain estrogen, progesterone both. These hormones prevent the pregnancy. The pill prevent sperm reaching cervix. Not here. The pill prevent ovulation. मतलब egg release होना बंद करते हैं. Then condom prevent sperm reaching the cervix region. Then vasectomy, semen contain no sperms, and copper T prevent implantation. Okay. So let me here achieving contraception, select their correct matching from the four options. So here the correct option is and prevent the pill that prevent ovulation, A3, condom that prevent sperm reaching the cervix region, vasectomy is permanent method and semen contain no sperms, copper T that prevent implantation. So how it is possible? Let me explain. So here the questions, the how pill is work, the mode of action of the pill, the patch, implants, novaring and Depo-Provera injection. So here pill, first pill inhibit ovulation. Pill inhibit ovulation. First, the ovulation due 14 days of the menstrual cycle, the ovulation occur. This ovulation means release of egg from the ovary. This ovulation process is prevented by the pill. First. Number 2, thickening of the cervical mucus. Cervical mucus means in cervix region where the sperm enter into the uterus, this region become thickening. So the passage or sperm passage is blocked. Number 3, prevent the normal build up of the endometrium. तो नॉर्मल जो एंडोमेट्रियम बिल्ड होता है जो इम्प्लांटेशन के लिए, दैट इज स्टॉप्ड बाय यूजिंग द पिल. दिस इज वेसोक्टमी वेयर द वेसा डिफरेंशिया आर कटिंग. यहाँ पे वेसा डिफरेंशिया काट दिया जाता है. सो स्पर्म आर प्रोड्यूस इन द टेस्टिस बट द स्पर्म डू नॉट ट्रांसफर. Okay, so only semen that produce in prostate gland, bulbourethral gland are transfer without sperm. Okay, then the copper T. Here copper T are used. Copper T are used in uterus. So here this prevent the transfer of sperm into the fallopian tube or egg transfer into the uterus. Okay. Number 2, this copper T secrete copper. Copper prevent the sperm motility. Number 1, copper stimulate the phagocytosis process that destroy the sperm inside the uterus. Okay. Next question is, a disease caused by an autosomal primary non-disjunction is. So what is autosomal? Autosomal means in our body there are two types of chromosomes are present. One is autosome and another is allosomes. Autosomes means 22 pairs that present in our body somatic cell, but in case of germ cell, egg or sperm, the germ cell, the sex chromosomes are present. So only one pair of sex chromosomes are present and 22 pairs of autosome are present. So here says a disease caused by an autosomal primary non-disjunction. Autosomal means disease caused by 22 pair of any of the 22 pair of autosomes. Non-disjunction. Non-disjunction means when the mitosis, during the anaphase of cell cycle, the chromosome are separate from each other and move towards the opposite pole. But here, due to some problem, either microtubule is not formed, the chromosomes do not separate from each other. The double number of chromosomes present in one cell and another cell remain empty. So in one cell, the number of chromosomes increase or double in number. That is called non-disjunction. So here let me explain Klinefelter syndrome. So what is the cause of Klinefelter syndrome? Klinefelter syndrome means extra Y chromosomes or extra X chromosomes are present in human body, especially in male category, where the chromosome number become X, Y or X, Y, Y. So chromosome number become 47. This is called Klinefelter syndrome. The others category, example, others category which looks like male but behave like female, this is called Klinefelter syndrome. Number 2 is Turner syndrome. Turner syndrome occur in female where number of X chromosome is decreased, but only one X chromosome is present. So chromosome number become 45. Means this is 2n minus 1 monosomy and he is 2n plus 1, that is trisomy. Example. Number 3, sickle cell anemia. So what is the cause of sickle cell anemia? Where in beta globin chain only one amino acid valine convert into glutamine. Means here GAG, here Valine means GAG convert into glutamine, that is GTG. Here's, let me explain. GAG means here the DNA level it is present, means you have a C, T, C and C, A, C. This is DNA level. The when transcription occur, then produce G, A, G, and you have here produce G, U, G. Okay. Then this produce valine and this produce glutamine. Here only 1 point mutation, here only this A and T are exchanged, that is called point mutation. So this due to this point mutation, the RBC shape become look like this, where the RBC normally shape is biconcave shape. So what happened here, the oxygen carrying capacity of the hemoglobin become decreased. So severe anemia occur in patient's body. The right answer is Down syndrome. So in case of Down syndrome, the 21 number of chromosome become double. Duplicate of 21 number of chromosome. So here the chromosome number become 47. It's occur in case of child. Okay. Then next question, pick out the correct statement. Hemophilia is a sex-linked recessive disease. The hemophilia means where the blood clotting factor is not produced. So if कहीं पर अगर कट जाएगा तो वहाँ से ब्लड कंटिन्यूअसली होता रहेगा, that is called hemophilia. Okay. This royal disease, this is also called royal disease. The hemophilia is a X-linked, X-linked recessive disorder. X-linked. Sex-linked मतलब when the disease are related to X chromosome. Down syndrome is due to aneuploidy. Aneuploidy means when the chromosome number increase or decrease. So in case of Down syndrome, the chromosome number become 47. 21 number of chromosome is duplicate, on duplicate copy of 21 number chromosome. So the total number of chromosome become 47. Or this is aneuploidy. That means the number of ploidy means chromosome number. Aneuploidy means increase or decrease. If increase, that is called hyperploidy, and decrease is called hypoploidy. So it is hyperploidy. Phenylketonuria is an autosomal recessive disorder. Phenylketonuria mainly cause phenylketonuria is when the normal क्या होता है, phenylalanine is a amino acid. Phenylalanine hydroxylase PHA, phenylalanine hydroxylase enzyme के द्वारा it convert into tyrosine. Then tyrosine again produce melanin, the pigment of our skin, hair, nail. Or so if the this mutation, if this enzyme is mutated or become inactive, then the phenylalanine do not convert into tyrosine. So phenylalanine deposited in the blood. Okay. So when urine is passes, the urine become musty odor. Okay. And the skin color, hair color become pale yellow, and it's prevent the development of mental ability. And sickle cell anemia is a X-linked recessive disorder. Not sickle cell anemia is caused by autosomal recessive. It is autosomal recessive disorder that is occur by a point mutation where the valine convert into glutamine. Clear? So here the right answer is hemophilia is a sex-linked recessive disorder, Down syndrome due to aneuploidy, and phenylketonuria is an autosomal recessive gene order. So the right answer is B. Next question. Next question is match the term in column 1 with their description in column 2 and choose the correct option. The dominance means here one allele are dominant over the another allele. Dominant means co-dominance. Co means colleague. That is both the allele are dominant. Pleiotropy means here single gene maintain many characters, and polygenic inheritance means here many gene maintain single character. First, dominance means when in a heterozygous organisms only one allele express itself. Means looks, except for example T and T, this is dominant. Homozygous condition T and T, the heterozygous condition, and T and T, this mean pure tall, this mean hybrid tall, and this means pure dwarf character. So here capital T means tall, small t means dwarf, but here small t do not express. In heterozygous condition small t do not express, that means t is expressed, tall is expressed. So here t is the dominant allele and t is the recessive allele. And codominance means like Mn blood group, here both the allele are expressed like blood group and Nn blood group. So when they express, both M and N are expressed here. Or AB blood group, both are codominant example. Pleiotropy means when single gene contain many characters, where the single gene influence many characters, like phenylketonuria. Here single gene or mutation of phenylalanine hydroxylase enzymes, it cause several type of problem, like increase the blood phenylalanine level, it do not produce melanin, this means skin pigmentation become decreased. Number 3, the brain development is harassed, do not properly develop, etc. When polygenic inheritance means many genes govern a single characters, many different type of genes govern single character, like human height, human body color, etc. both are polygenic inheritance. So right answer is dominance. Right answer is dominance means heterozygous condition, both only one allele express itself, like it A2. So A2. Codominance means both the allele express, B3. So A2 and B3. D. Pleiotropy means a single gene influence many characters, C4. And D. Polygenic means many genes govern a single character. So here D is a right answer. Next question. A man and woman who do not show any apparent signs of a certain inherited disease have 7 children, 2 daughters and 5 sons. 3 of the sons suffer from the given disease, but none of the daughters affected. Which of the following mode of inheritance do you suggest? So here, like this is mother and this is father. And 7 children, 2 daughter and 5 son. No, 5 sons. Then the sons suffer from the 3 sons, means 1, 2, 3. 3 sons are affected and 2 sons are normal, and these 2 girls are normal. So here, in case of sex-linked disease, sex means X-linked disease. So here mother is X dot 2XX and father is XY. So here father transfer his X chromosome to daughter, means here one X chromosome and another X chromosome, that is called XX or XX. In case of daughter, one X chromosome come from father and another X chromosome come from mother. But in case of mother, the X chromosome come in both daughter and son. So if the father X chromosome is affected, then the daughters become carrier. But if mothers is affected means when both the X chromosome are affected, then 50% daughters are affected, 50 daughters become carrier, and 50% and all the males are affected. But here some males are normal. That means mother is a carrier. Only one X chromosome of mother is affected. So when the mother transfer this X chromosome to boy, they affected. But when mother transfer this normal X chromosome to son, then it is not affected. So here the genotype is XY. Here X normal, but here X is one X is affected. Okay. So here this is the and also, and in case of sex-linked disease, mostly males are affected. Rare number of females are affected because females have 2 X chromosome, males have only 1 X chromosome. So here the right answer is sex-linked recessive disorder. Okay, sex-linked recessive disorder. The next question. If there are 999 bases in an RNA that code for protein with 333 amino acid, so one amino acid here, let me explain, one amino acid that carry 3 nitrogen base, okay, like A, U, G, methionine. So here 999 bases are present. So 3 base contain 1 amino acid, so it is divided by 3, and here 3 amino acid are produced. So from 999, 333 amino acid are produced, and the base position of 901 is deleted. So here suppose 1 to 901 to 999. Total amino acid, total base 999. So 901 is deleted. So 1 to 90 are normal. Here 90 to 999. That means 99 amino acid are altered. Here 901 position is deleted. So deleted such that the length of the RNA become 998. So it become, when the 901 position is deleted, it become 998. Means here 98 amino acid are produced, altered. So 98 bases means 332 point something. Okay, that means 33 is the answer. So here the right answer is 33. Next question. Select the 2 correct statement out of the 4 statement given below about lack operon. So what is lack operon? Lack operon means in the presence of lactose, the structural gene are expressed. How they expressed? In the presence or absence of lactose in prokaryotic cell. So here glucose or galactose may bind with the repressor and inactivate it. In the absence of lactose, the repressor binds with the operator region. The Z gene codes for permease. This is elucidated by Francis Jacob and Jacob Monod. So this is operon. This is operon. The ZYA is structural gene, and here O for operator region, P for promoter region, I for inhibitor or repressor protein, and P for catabolic activator protein. P for cap. P means here P represent cap, catabolic activator protein that stimulate this transcription process. So first Z means beta galactosidase enzymes, Y means permease, and A means transacetylase. So here the option, the Z gene codes for permease, not Z gene code for transacetylase enzyme. So here 3 is not correct. 3 option is not correct. Number 2, this was elucidated by Francis Jacob and Jacob Monod. Yes, Jacob and Monod first proposed this operand model in prokaryotic cell in 1953. In the absence of lactose, the repressor binds with the operator region. So here look, this is the inhibitor protein, inhibitor gene that by transcription and translation process produce a repressor protein. This is when lactose is absent, when the lactose is absent, then this repressor protein combine with operator region, and this operator region is blocked. So RNA polymerase enzyme 2 that bind with the promoter region do not move towards the structural gene. So no transcription occur, no product are found. These genes are not expressed. But when the lactose is present, lactose means in lactose is called inducer. So this operon is called inducible operon. So here lactose bind with the repressor and the repressor become inactivate. So the repressor do not bind with the operator region, and when this do not bind the operator region, the RNA polymerase present in P region or promoter region easily move from here to this region. So the transcription and translation of Z, Y, A structural gene occur. So this product are found. Clear? So here right answer is the glucose absent. Here the right answer is in the absence of lactose, the repressor binds with the operator region. Right? Lactose when absent, repressor bind operator region. This is the correct answer. Jacob Monod discovered correct answer. Glucose or galactose may bind with the repressor. Here glucose or galactose is not necessary. Here lactose, this is lack operon, so lactose is necessary. The right answer is 2 and 4. 2 and 4 means this C portion is the right answer. Next question. During translation, initiation in prokaryotes, a GTP molecule is needed in formation of formyl methionine tRNA, binding of 30S subunit of ribosome with mRNA, association of 30S mRNA with formyl methionine tRNA, association of 50S subunit of ribosome with initiation complex. तो यहाँ पे बोल रहा है कि when the translation initiation occur, at the initiation states, during translation the GTP is required as a source of energy. When formation of formyl methionine tRNA, because initiation this formation means the methionine tRNA are formylated. Okay, methionine attached to the formylated methionine attached to the tRNA region portion. Here no energy is required. Binding of 30S subunit of ribosome with mRNA. When the 30S ribosome to ribosome produced by 2 subunit, 30S ribosomal subunit and 50S ribosomal subunit, that is 30S and this is 50S. And association of 30S mRNA, when this is suppose this is mRNA, and first this is 5' region and this is 3' region. So first is signed Dalgarno sequence are present. So here IF, here 30th subunit of the ribosome are attached, IF3 with the initiation factor 3 and initiation factor 1. Clear? Then the tRNA, this is formaldehyde methionine tRNA, F met tRNA are come here start codon AUG present. The opposite base U A C are here formatted methionine tRNA. When it carried into this region by IF2, I initiation factor 2, it use GTP into GDP phosphate, and this phosphate, so phosphate is released. This phosphate is required to binding the formatted methionine tRNA to this region. So when the when the association of 30th RNA with formatted methionine tRNA, this is the answer. 11 number, this is the answer. Let me explain properly. This is the translation of initiation site, initiation process. Here this is 30th ribosomal subunit of the RNA or ribosome. This is with the help of some initiation factor 1 and initiation factor 3, they are combined and a signed Dalgarno sequence of the mRNA. This is mRNA. Then here AUG is present. The AUG start codon methionine. So in case of prokaryotes, formaldehyde methionine group, methionine here methionine plus formyl, methionine plus formyl group are added. But in case of eukaryotes, this formaldehyde methionine are not present. In case of eukaryotes, only methionine tRNA are present. But in case of prokaryotes, methionine formaldehyde tRNA are produced are required. So this formaldehyde methionine tRNA are bind to AUG region, start codon region. So and this is by IF2. So here GTP is required to attach this formaldehyde methionine tRNA to AUG region. So right answer is association of 30A subunit mRNA with formaldehyde methionine tRNA. Next question. In the genetic dictionary, there are 64 codons as 64 amino acid. Total number of amino acid are 20. Total number of amino acid are 20. This 20 amino acid is coded by 4 nitrogen base. Okay, coded by 4 nitrogen base, and there are 4 nitrogen base. What is this? Adenine, guanine, cytosine and uracil. Okay, so here 4 to the power n or 4 to the power 3. 3 is the number of nitrogen base and 4 total number of nitrogen base, and for one amino acid 3 nitrogen is required, means 64 code. So for 20 amino acid there are 64 code are required. 64 amino acid are to be coded? No, only 20 amino acid are coded. 64 type of tRNA are present, but there are many type of tRNA are present. There are 44 nonsense codon and there are only 3 nonsense codon: UAA, UAG, UGA. Ochre, amber and opal. These are called stop codon. Stop codon. This codon stop the translation process. It is not carry any information or any amino acid. So it is called nonsense codon. So here only 3 nonsense codons are present and 61 codons are sense. Genetic code is triplet. So all genetic codes is triplet. So AUG, 3 nitrogen base. Okay. Next question. In the genetic, this is the genetic code, total 64 codon. These 3 are non-genetic code, means they do not carry any amino acid information. This is called stop codon or nonsense codon, and the remaining codons represent amino acid. So here sometimes one amino acid carry one or two or many codons. Okay, here suppose valine contain 4 amino acid, 4 codon. Okay, next, the chronological order of human evolution. The human evolution का chronological order है क्या? Australopithecus, Ramapithecus, Homo habilis, Homo erectus. Ramapithecus, Australopithecus, Homo habilis, Homo erectus. Ramapithecus, Homo habilis, Australopithecus, Homo erectus. Australopithecus, Homo habilis, Ramapithecus, Homo erectus. Here the right answer is Ramapithecus, then Australopithecus, then Homo habilis, Homo erectus. This Ramapithecus is the common ancestor of monkey, chimpanzee, ape and human. So let me explain. This is the chronological order of human evolution, where first 15 million years ago the Diopithecus are occur. Then Ramapithecus 2 million years ago, Australopithecus, Homo habilis, then Homo erectus, then Homo neanderthalensis, then Cro-Magnon and Homo sapiens, and modern man Homo sapiens sapiens. This is the chronological order of human evolution. Next question. The eye of octopus and eye of cat show different pattern of structure, yet they perform similar function. But structure are different but function are similar. Structure are different but function are similar. It means structure different means this is convergent type of evolution. Okay, means suppose bat, suppose insect and bat. Okay, this is derived from arthropoda, derived from mammals, but both have winged for the flying. The structure are different, but they do same function for flying. Same function, fly. Okay, this is called convergent evolution. Convergent evolution, and it is homologous organ. Sorry, analogous organ. Analogous means different structure. But when, like bat, like human, like whale, they have common ancestor, that is mammals. Okay, then what happened? They live in different place, different environmental condition, and in different environmental condition they adapt differently. Like bat produce potassium for flying, human have leg for walking or running, and whale have paddle. So here the divergent means ancestors are same and they live in different place, so they adapt differentially. Okay, this is called divergent evolution. Divergent evolution. This is called homologous organ. Homo means structure, paddle structure or potassium structure or human limbs structure are same. Structure are same. Number of bones are same, but adapt differentially. Homologous organ. So here the eye of the cat is mammals, and eye of the octopus. Okay, octopus means they are mollusca. So mollusca and cat, mammals, they are different ancestor, but so structure different but function are same. This is analogous organ that have evolved due to convergent evolution. Analogous means organ are different but evolved they are same, function are same, that is convergent evolution. Some other example, thorns of bougainvillea and tendrils of cucurbita. Thorns of bougainvillea and tendrils of cucurbita. Their structure different, but their function convergent, means their function same but structure different. Source different, source are different. Okay. And wings of butterfly and birds. Okay. Their ancestors are different but function are same. The right answer is A. Next question. 15. Which of the following is the correct group of vestigial organ in human? Vestigial organ means some organ are active, organs are active in past, but now they are inactive. Okay. Say for example, nictitating membrane in our eyes, the eye corner, beside the nose, the eye corner, some muscles are present, some membranes are present. If you see, when the birds close their eyes, a thin membrane covers their eye. And open the eye, then membrane remove. But in case of human, there is no membranes are appear. So this is called nictitating membrane. That may be active in past, but due to do not use or less use, this become inactive. Ear muscle, also eyelids and coccyx. So here wrong answer is eyelids. Our eyelids are active. Appendix, coccyx, ear muscle and elbow joint. No, here the wrong answer is elbow joint. Our elbow joint are active, not vestigial part. Wisdom teeth, coccyx, body hair and ear muscle. Wisdom teeth, coccyx, body hair, nictitating membrane and ear muscle. Here the right answer is wisdom teeth. Wisdom teeth means molar teeth, the last teeth of our mouth. Body hairs become, in case of monkey the hairs are present, but in case of human the hairs do not use, so they become vestigial part. Nictitating membrane and vermiform appendix. Vermiform appendix also the vestigial part of our body. The next question, match the disease with its correct type of vaccine. Vaccine means here some disease name and what type of microorganism are used in such cases. That is called that is represent which type of organisms, because what is vaccine? Vaccine is a killed microorganism. Vaccine is a killed or attenuated or inactive part or whole organism that is caused disease. Okay. So here tuberculosis Mycobacterium tuberculosis, that cause TB. These in such cases the killed bacteria are used. Whooping cough is a disease. Okay, it is harmless bacteria. Diphtheria, Corynebacterium diphtheria, inactivated toxin. Diphtheria toxin, Corynebacterium diphtheria produce one type of toxin. This toxin cause this disease. So we scientists collect the toxin and this toxin become inactivated in the laboratory. Then the inactivated toxin are injected in our body to promote or stimulate our immune system. And polio, this is polymyelitis virus. So here the polymyelitis virus are injected in our body in inactivate condition or harmless condition. Okay. When the microbes are cultivated in different pH condition or different temperature, their disease causing ability become decreased. Okay. When then these microorganisms are injected in our body, these microorganisms become inactive, but our body identify these microorganisms as an antigen, and our body produce different memory cell and huge number of antibody against this pathogen. This is a strategy of vaccine production. Okay. Next question. So here tuberculosis is a killed bacteria, whooping cough harmless bacteria, diphtheria inactivated toxin, and polio is a harmless virus are used. Here are some secondary alkaloids and their source and use. The first, cocaine. Cocaine produced from erythroxylon coca. Cocaine produced from erythroxylon coca. Accelerate the transport of dopamine. Cocaine mainly accelerate the transport of dopamine that stimulate the release of stimulate the brain to release the energy hormone norepinephrine. Heroin is cannabis sativa, not. Heroin is obtained from Papaver somniferum or poppy plant. This source is wrong. Depression and slow down the body function. It is a depressant, means it inactivate our nervous system, so body become relaxed, slow down the body function. Cannabinoid, this is also wrong answer. It is from cannabis sativa, canna plant, produce hallucination and hallucination, right? Hallucination, murcha jana. Morphine, morphine from Papaver somniferum and is sedative and painkiller. This is sedative and painkiller. During the surgery, this medicine is used as sedative and painkiller. So here correct answer is D and correct answer is cocaine. Two answers correct. The main reason why antibodies could not solve all the problem of bacterial disease. Suppose you have a bacterial disease and doctor prescribe you antibiotic. You use this antibiotic long time. Okay. And when you use a antibiotic against a similar bacteria, the bacteria slowly adjust this antibiotic or adapt this antibiotic due to the mutation of bacteria. Then these antibiotics do not work against this bacteria. So here the correct answer is development of mutant strain resistant to antibodies. Nowadays in medical science, the most dangerous threat of human being is to resistance of bacteria against antibiotic, because medical company produce many type of antibiotic, and this against this antibiotic bacteria also change their habitat. They adapt this type of antibiotic, so these antibiotics do not work against bacteria. So again new antibiotic are produced, okay, by medicine company. Next question, which of the following enhance or induce fusion of protoplast? The first, what is protoplast? Protoplast is the cell without cell wall. The plant cell wall is made by cellulose, pectin, hemicellulose, lignin. Then the first collect a cell and these cells are treated with different type of enzymes like cellulase, hemicellulase, ligninase, okay, pectinase. So cell wall is removed. The naked cell is called protoplast, because cell wall, when the cell wall removed, the cell membrane exposed. The cell, when the two protoplasts are fused in the presence of polyethylene glycol and sodium nitrate, these two protoplasts, cell membrane of the protoplast, are fused and the cytoplasm are mixed. Okay. Then the cytoplasm, after mixing of cytoplasm, the two dikaryon condition are formed. This dikaryon condition, the nucleus are fused, nuclear fusion occur, and new hybrid plants are produced. Okay. This is called the, so here example is polyethylene glycol and sodium nitrate accelerate the fusion of two protoplasts. Next question or last question is, consider the following 4 measures that could be taken to successfully grow chickpea in an area where bacterial blight disease is common. So what is the bacterial blight disease? The disease, blight means burnout. The leaf, after burning of the leaf, burning के बाद leaf जैसा दिखता है, ठीक उसी तरह after infection by bacteria, the leaf become blighted. Okay. So to recover this disease, you can use first some chemical that kills the bacteria. Number 2, resistance variety. So here spray with Bordeaux mixture. Here spray with Bordeaux mixture. Bordeaux mixture means here use copper sulfate plus lime plus water. This is the mixture of Bordeaux mixture. Okay, so you can use the Bordeaux mixture. This is the right answer, and use the varieties resistant to diseases, because you would have only disease free seeds. But if you use disease free seeds, sometimes we plant healthy, but it is not sure that plant resist that type of disease. Control of insect vector of the disease pathogen is very hard process. Okay, so use a variety, resistant variety to the disease and spray with Bordeaux mixture. So this is the last question of our video. This is the end of this video. If you like this video, then please subscribe, like and subscribe my channel. Thank you.